Hello, and welcome to the 24th Florida Keys Mosquito Control District Oxitec Public Educational Webinar. Our topic is takeaways from 2023 and looking ahead. As presenters, we have Chad Huff. He is the Public Education and Information Officer for the Florida Keys Mosquito Control District. Rajiv Vajanathan is the Director of U.S. Programs for Oxitec, and I'm Meredith Finsom, Oxitec's Head of Public Affairs and a Florida native. We are hosting the series of public educational webinars to share information, especially with residents of the Florida Keys, and to provide forums to answer questions. These webinars are open to everyone. They are recorded and made available after the event. All questions relating to the webinar topics will be answered. If we receive a lot of similar questions, we may group them together. We will spend the second half of the webinar taking questions, but you can go ahead and start to enter your questions in the chat box now. If you are a Florida Keys resident, we give priority to your questions. So please list the key that you live on, and we will prioritize those questions. If we run out of time, you can send questions in writing to florida@oxitec.com, and we will answer them. Our agenda is to give a bit of a project background. We're going to review our takeaways from this year's project. It was the third season, the third year of the project. Talk a little bit about what might come next, and then we'll move on to our Q&A. Chad, over to you.
All right, Meredith, thank you, and thank you everybody for the opportunity to be here with you. A lot of people ask, especially my business as a public information officer for the district, why this project and why now and why in the Florida Keys? And really the answer is pretty simple. There is a definite need for it. Anybody that's spent any time in the Florida Keys or has lived with us down here knows that dengue fever, which is a mosquito-borne disease, has been an issue in the past. It was an issue in 2010, and then we had a local outbreak as recently as 2021 and 2022. In Key Largo, we had about 70 people. I think it was maybe a little less than that had locally acquired dengue. And in mosquito control, any mosquito-borne illness is too much, but when it's locally acquired, it really gets your attention because it means it's being actively transmitted on the ground in your area with mosquitoes. So dengue fever is also known as breakbone fever. It's very prevalent in third world countries, but by and large, it hasn't had much of a foothold in the United States. And that's thanks in a large part due to mosquito control. And it's certainly something that gets our attention whenever it shows back up. There's also a threat of other diseases, diseases with scary names like chikungunya and Zika. Yellow fever certainly has had a history in the Florida Keys, given our shipping traffic and our proximity to the Caribbean. We've had a history with those things. So it's something that is just kind of part of life down here, and we are always on guard for. One thing about mosquito-borne disease is you rely on your traditional mosquito control methods to control those mosquitoes. And anybody who's spent any time researching mosquito control knows that some of those methods aren't working as well as they once did. Pesticide resistance is a problem. It's industry-wide. It's not just in the Keys, but everywhere. And so good mosquito control organizations are always looking for projects that tick that box that would control mosquitoes without relying on pesticides. And then there's obviously an ecological benefit. Mosquito control in 2023 uses ultra low volumes of pesticides, so we feel like we are as responsible as we can be when it comes to the environment. But obviously a project or like the Oxitec product ticks a box to where it is environmentally responsible as well. So a lot of those reasons are why we've been talking to Oxitec for years about this project, and why we've been involved in it for the past 3 years very successfully thus far. Go ahead, Meredith. Dengue fever, you'll hear us talk a lot about that, and it's because it's getting a lot of headlines in Florida, and not just in the Keys. Like I said, in 2022, there were 68 locally acquired cases, 750 travel-related cases of dengue now reported in South Florida. So far, not in the Florida Keys. This is in South Florida beyond here. A lot of those cases in Miami-Dade and Broward and in Palm Beach County. So far this year, the tide hasn't really slowed. In fact, it's sped up. 98 cases of locally acquired dengue reported in Broward, Hardy, Miami-Dade, and Palm Beach counties, and more than 400 travel-related cases. A lot of that is attributed to dengue that has upticked in the Caribbean and in a lot of other places. It's just it's a very, very prolific disease, and mosquito control in the Florida Keys, just because of our nature with tourism and because of our proximity to the Caribbean, it's something we're always on guard for. But for the last couple of years, it's really been prolific just to our north, and now in parts of the state, all over the state. Go ahead, Meredith. So Aedes aegypti, it's a mosquito you'll hear us talk about. I don't know that I can ever remember hearing or acknowledging an Aedes aegypti mosquito before I began working in mosquito control. But I think now, as a parent and certainly working in mosquito control, that is a mosquito that would have my attention. The Aedes aegypti is not native to Florida. It is not native to the Keys, but it vectors almost every single disease case that comes into the Keys that you hear about. It is vectored by the Aedes aegypti mosquito. And this graphic here kind of shows that the Aedes aegypti has done a very dutiful job of marching across the world and getting into places where it is not native, and then getting a foothold, and then from there being a threat. And people who don't know about Aedes aegypti in places like California, where they didn't have it that much in the early 2000s, are now hearing an awful lot about it for this very reason. They are just a very good mosquito that lives around people. It bites people almost exclusively, and it lives in containers and in things that we as people discard. So it is a very symbiotic sort of relationship. Wherever people goes, the Aedes aegypti is soon to follow, and unfortunately, more and more people are becoming sick as a result. One thing that we want to mention now is malaria. This was something that we started hearing about in Florida this year. It was several hundred miles away from this project, had nothing to do with this project, but it gets a lot of attention because a lot of people have relatives that dealt with malaria in the past, and it was really a disease that you didn't hear much about. It was one that it's kind of like polio. You heard stories about it, how bad it can be and how terrible it can be. And when somebody turns up with it now, you really notice. Well, malaria this year, there turned out to be 8 locally transmitted cases of malaria about 400 miles to our north here. And it's kind of the exclamation mark on where we are with mosquito control and disease control these days. So the need for novel methods has never been larger, and that's kind of what got us here and into our third year of the Oxitec project that we're glad to be a part of. And I think we've got some pretty promising results as a result of our association, and we look forward to everything that comes from that. So thank you very much. I'll stand by for questions. On to you, Rajiv.
Let's see, I'm going to unmute, and I think we're good. Thank you, Chad. Thanks, Meredith, for that introduction. I'm going to present a little bit about our technology and what we've done in Florida, largely for 2023, but it also includes a lot of what we've done in 2022 as well. In a way, almost agnostic of what insect we're talking about, because we use the same platform technology for mosquitoes and also for agricultural pests. But if we're going to talk about mosquitoes in particular, we'll use this as a model. This is a male Aedes aegypti that we see on this slide. And the males of any mosquito don't bite. Only female mosquitoes bite. Male mosquitoes do not penetrate skin, and they're incapable of drawing blood. Our male mosquitoes and our female mosquitoes have two genes that we have inserted into them. One is a self-limiting gene that very specifically builds up in the larvae of females, resulting in female-specific lethality. And connected to that self-limiting gene, which is specific for females, we also have a fluorescent marker gene. And so we can tell by collecting larvae and pupae in adults if this is one of ours or if it's one of the progeny of ours, because we can look at them under a red filter on the microscope and they'll fluoresce red. While they may be morphologically indistinguishable from any other Aedes aegypti, we know that if we look at them under the microscope with a red filter, we can tell which one is carrying this gene. And if it's carrying the marker gene, the fluorescent gene, then we know it's also carrying the self-limiting gene. Both the proteins for the marker, the fluorescent protein, and the self-limiting gene are non-toxic. They have no effect on humans, frogs, fish, other invertebrates that might be in these aquatic habitats. Specifically, because this is effective in female larvae, anything that comes out of our boxes are males only, male Aedes aegypti. And I'll go into 3 lines of evidence that show that we're only releasing males and that only males survive. Our males are traceable in the field because of the fluorescent marker gene, and they are self-limiting because we are not constantly releasing them. So we do release for a season, and then we stop. There is a cessation of that, and then we collect eggs and adults for weeks following. And we find that there is a decrease in the frequency of the marker gene in the population. So we can continue. I'll talk about what our plan was for 2023, what we accomplished, and this is evaluating our male mosquitoes in the Florida Keys. Meredith, thanks. For 2022 and for 2023, so it's kind of a replicate. Our design was very similar. So for both 22 and for 23, we had 2 pilot projects, one a wide scale for entire neighborhoods, and another one for individual homeowners. We call them Project B and Project D. Project B is on a neighborhood level. So we don't need to have a release site at everyone's home. And if you've ever been to the Florida Keys, you would also know that that's impossible because about half the homes are actually rentals. So you're never going to get house after house after house. All we really needed were one host for about 1 hectare or 2.4 acres. So we could go into a neighborhood, knock on doors, talk to people and say, do I have your permission to set this up? And we had overwhelming support. If you think about it, there is no way we could have done this in 2023 if we did not have neighborhood and community support in 2022 and in 21. And so this large neighborhood release is really to look at suppression values and to see how well our males mate, what kind of coverage we can get on a neighborhood. And this is really a tool for mosquito control districts or for homeowner associations. So it's a large-scale, neighborhood-wide release of our males. Project D, on the other hand, is with the individual homeowner as an end user. And so if one person wants to have a box in one's backyard and release, we wanted to collect in that one area. So around around one yard and possibly adjacent neighbors. We wanted to know how well this can be used by the homeowner. So while both have similar endpoints, we are getting different metrics, right? So for the neighborhood scale, we want to know how well can we suppress on a wide scale. And for the Project D or household scale, we wanted to know how well are our boys flying, how far are they flying, how well are they mating. And we'll talk about some of those metrics in the next few slides. For our 2023 releases specifically, we wanted to show strong performance of our males. And so by performance, it's really how well are they mating and how are we tracing that using the fluorescent marker, how far are they flying, and how long are they living. We also wanted to include data on mating performance for these small areas or the single properties or Project D. And all of this, right, everything we've done in 21, 22, and 23 is really to put together a dossier that we submit to the Environmental Protection Agency or EPA or to the Florida Department of Agriculture and Consumer Services or FDACS with the goal of commercializing this product. So the goal is really, to move from thinking about this as a project to a product. How might we use this at the level of MCDs, that is mosquito control districts, or HOAs, and how feasible is this as a product for someone to just use at home? A little bit about the logistics. Our Project D or neighborhood-wide releases started in April on Vaca Key only. And so there were three sites or three neighborhoods on Vaca Key where we released our male mosquitoes. So that means a lot of footwork and knocking on a lot of doors and asking people if we have their permission to, one, collect mosquitoes, and two, to release our male mosquitoes. And as I said, there is no way we could have done this if we did not have community-wide support. Very proud and really humble at the same time for the support we've gotten in the Keys. Those are our three treatment sites on VACA. We also had 3 control sites, that is untreated, no male releases, only surveillance. And those were also largely on VACA with 1 or 2 on Key Colony Beach. Separately for Project D, the individual homes, we had 11 individual homes, and we had to spread these out. They're just, kind of limited by geography. So we certainly had the majority of our treatment sites for Project B on VACA, and also on Fat Deer, Grassy, and Duck Keys. At the same time, we had 11 untreated sites that we could compare our treated sites to, also on VACA, Conch Key, Duck, and also on Key Colony Beach. And again, thank you to our box hosts and our egg trap hosts and our adult trapping hosts, without whom we could not have done this. How do we do it? Right? People, first question they ask when we knock on their door, how do you collect mosquitoes? So we do it two ways at all of our sites. Mosquito females lay their eggs either on water or on a moist substrate. All mosquitoes in the genus Aedes, so not true for Culex, not true for Anopheles, but all mosquitoes in the genus Aedes lay their eggs on a wet substrate, not on the water itself. So we put these little black cups with a zip tie. We might attach them to a fence or the base of an areca palm or bamboo, and we put a tongue depressor, so a wooden tongue depressor, into that, and half fill it with water so the water wicks up the tongue depressor. Then we go in with our phones, and as we remove it, read it. Each one has a QR code, so we know who's read it, at what time, on what day, and it has a GPS location. So we can take that back into the lab. So we check those tongue depressors, or what we call egg paddles. We check them for eggs. We look at them under the microscope, and eggs, mosquito eggs of Aedes, look like very, very small black grains of rice. So we count the number of eggs. And we use that, one, just to get a baseline idea of how many, what your pressure is for Aedes aegypti. And when we bring it in, we then put those egg paddles or tongue depressors on water. The eggs hatch. So now we have an idea how many larvae we have. So we have total number. And then we look at that under a red filter. So now we know what the mating success is. In other words, what percentage of all those Aedes aegypti larvae are sired by our males, because they'll be fluorescent. So that's all from the larval perspective. From the adult perspective, the technology for collecting mosquitoes, no matter how sophisticated it is, it still has about 4 simple components. And that is, it has a light, it has a fan, it has a mesh bag underneath, and all of this is hooked up to a battery. And so mosquitoes are attracted to the light, they're sucked down by the fan, and they end up in a mesh bag. So now we can go in regularly, remove the mesh bag, freeze it, killing the mosquitoes, and put a new mesh bag in. And so that way, we're going to collect a lot of mosquitoes. And some of them might be Culex. Many of them are Aedes taeniorhynchus, the salt marsh mosquito, which is common in the Florida Keys. But we also collect Aedes aegypti. So we use these 2 tools, collecting eggs, bringing them in, letting them hatch, and then counting the larvae, and at the same time, collecting adults. And in a way, again, agnostic of what that mosquito species might be, just collecting a lot of mosquitoes, bringing them into the lab, freezing the bag, and then, as you can see in the slide, looking at them under the scope to identify the mosquito species, and then differentiate between male and female. And then when we have Aedes aegypti, looking at them under the red filter to identify which ones are either ours or sired by our males. So let's get to the results. Again, just to reiterate, thank you. We couldn't have done this without you. Well, first of all, we could not have done this without the Florida Keys Mosquito Control District, FKMCD, and we could not have done this without the support and real encouragement of homeowners and neighbors in the Florida Keys. So thank you. Couldn't have done this without you. We've been through some storms in 2021 and 2022. We've been through some pretty severe tropical storms. This year, Fortunately, we made it through. And that's a big deal because we did not have to pause and restart. So when we talk about the neighborhoods, the Project B specifically, this year we started in March and we ended in October, just to coincide, of course, with warm weather and warm, rainy weather. And we found that our males flew just as far as any of the wild type males that you would ordinarily find. So the best thing I can say about our results regarding male longevity and male flight radius is that there's nothing interesting. They live as long as the wild type males, and they fly as far as the local males, the pest species. So that's boring, and boring is good, that they live as long as anything else. We also found that in these areas, the Project B areas, that our Oxitec males found and mated successfully with the local females. We know that because we collected eggs from those egg cups. We removed those egg paddles. We brought them into the lab, put them in water, and hatched out the eggs. So we have an idea what the pest pressure is in these neighborhoods, and we have an idea what proportion are fluorescent. And so they carry the marker gene, which is connected to the female-specific lethality gene. And here's the biggest one. Really proud of this, and I'll tell you how we got here. 100% of the female larvae that carried our gene died. And I'll tell you how we know that. One, going back to the different ways of collection, we collected eggs, right? We brought the eggs into the lab and hatched them out. And now everything looks the same when you don't have a red filter. When you look at it under red filter, you can count out how many of them carry the target gene. So we remove those from the samples and we put them in a cup. We allow the larvae to develop, pupate, undergo metamorphosis, just like a caterpillar to a chrysalis to an adult butterfly, and then emerge as an adult. And we've looked at over 60,000 of these. Every single one emerged as an adult male. There is no ambiguity that when they're carrying the gene, that it results in a female-specific lethality, and that the adults only emerge as males. So that's one line of evidence by collecting eggs, taking them to larvae, and then letting those larvae pupate and emerge as adults. Line number two, whenever we put those boxes out, we'll always put out one, and then we'll put out a proxy, same water, same egg batch, same food, maybe about a meter or two from the first. But the proxy box we put inside a butterfly cage or a bug dorm, as they're called, right? So this is what kids use to raise caterpillars to butterflies. It just traps everything inside. So this enables us to do two things. One, we can count what our release rate is. If we just put a box out and say, trust me, this is how many come out, well, we don't know that. But if we put two of them out and we have proxy boxes for our release boxes, then we can go in Monday, Wednesday, Friday with a handheld vacuum cleaner and collect the mosquitoes that have emerged. Having done this now for all of 2021, 22, and 23, not a single one of our adult emergence from the release boxes was female. So we know that everything we're actually putting out is male. That's our second line of evidence. Our third line of evidence is, remember those mosquito traps I mentioned? The ones with the light, the fan, and the mesh bag? So those are great for baseline. It helps us look at population trends, but it also tells us what we're collecting, because when we have a proxy box, that tells us what we're releasing. when we collect with one of these light traps, it tells us what's in that environment. So we collected a lot of mosquitoes. Some of them, many of them, were Aedes taeniorhynchus, the salt marsh mosquito. We collected a lot of Aedes aegypti as well, and we certainly collected male and female Aedes aegypti. But when you look at them under the scope, every single adult Aedes aegypti was male. We did not detect a single female Aedes aegypti using the light traps. And so with these three independent lines of evidence, collecting eggs, hatching, bringing them up, none of those emerged as females. Getting a proxy box next to our release box and vacuuming everything that comes out, all male. And when we use the light traps and collect a lot of mosquitoes, those that were fluorescent, i.e. carried the tracker gene, all male. So we know without any ambiguity that our construct works, that we are releasing males, that they're mating with local females, and when those females lay eggs, their male progeny survive and emerge and go on and mate, but their female progeny die as larvae. The consequence of this is that we have reduced local pest populations of Aedes aegypti in areas where we saturated with our adult males. And one thing we did not expect, but we were really excited when FKMCD shared their data with us, was we wanted to ensure that we were not selecting for behavior where mated females laid their eggs in these black cups. So we asked FKMCD if they would collect eggs or larvae from artificial sources, natural and artificial water sources like gutters and buckets, wheelbarrows, children's toys, and from a surprising number of jet skis and upside-down kayaks where rainwater had collected. Our FKMCD partners provided us with thousands of larvae, Aedes aegypti larvae, from all of these other habitations around people's homes. So we could do the same thing with these collections that we did with our egg cups, namely, look at them under the scope and count them. Still no differentiation. Then look at them under a red scope and see which ones are fluorescent. Remove those, put them in a separate cup, and then allow them to undergo metamorphosis, pupate, and emerge. Every single fluorescent larva emerged as an adult male. This was really good news because what it tells us, as Chad alluded to earlier, Aedes aegypti females lay their eggs in what we call cryptic or hidden water sources. A bucket might be obvious, a wheelbarrow might be obvious, but 1,000 bromeliads are difficult to check. And Aedes aegypti will lay their eggs inside bromeliads, inside the bracts of pineapple-like plants. It's hard to get in there. But what this indicates is that our gene can be used to control Aedes aegypti in A, places that are non-obvious, and B, places that a mosquito control district might not have the authority to actually enter or the technical ability to treat. It's hard getting an insecticide above and around and behind a building, and a mosquito control district cannot treat there if they don't know it's there. So in a way, this is the female Aedes aegypti helping us, doing our job for us, and carrying that female-specific lethality for the larvae in places that a mosquito control district might not see and might not have access to. Separately, for Project D, recall, those are for individual homes in a larger neighborhood. Our treatment for Project D overlapped with Project B, so that was about April to October. We staggered it for ourselves because it was a lot to do. And I'm going to try to finish up soon. And we found that our smaller boxes also produced males. They produced fewer males as we expected. We found that our males dispersed as we expected and that they mated with local Aedes aegypti females. Again, the real take-home message, Project D, as for Project B, is that every single female larva that we collected died as a larva, and we had 0 female adults emerge. Just go into a little more detail, tell the story here, Meredith, and I do want to end up soon. So some of our take-home messages. 1, we know that our males mate at parity with the local males with the females. So the fitness of our males, their longevity, and their flight radius is at the same capability as the local males. We seem to have come back. There we go. Oops. And that we found 100% female-specific larvicidal efficacy. So all of our female offspring from our released males died in the box. The box that you're looking at, that's the mini box, and the pro box is larger, about the size of a basketball, and demonstrating female-specific larvicidal efficacy against 1 species of mosquito, Aedes aegypti, a non-native invasive species that is an important vector. And that these results from 2023 are consistent with 2022. Yay! So we had a good replication of data for 2 years in a row in 3 different project sites, and that they reiterate our results from previous years in the US and with our ongoing work in Brazil. Next one. So now we've stopped. We are not releasing any more males at any sites. We stopped in last week of October. So now it's all about surveillance. We are still setting up our oviposition or egg cups and still collecting our egg paddles. And we are still collecting with the light traps. And we will continue to collect even though we have stopped our releases. And that is to monitor fluorescence in larvae and in adults. How long is it going to persist? In years prior, it does persist for about 2 months, but it's hard to say because it depends on rain and on irrigation. Recall Aedes aegypti eggs can remain dormant for months, which is remarkable, until they're flooded, either by rain or by irrigation, by watering a pot and filling up the ceramic saucer at the bottom. So we are collecting data, and we have from 21, 22, 23. This monitoring and surveillance will continue into 2024 so that we can say that our frequency of fluorescence decreases from week to week until we cannot detect it anymore. That's why we call it a self-limiting gene. This gene does not amplify in the population. Once the homeowner or the mosquito control district stops their releases, there will be an attrition in the degree of fluorescence and hence in the efficacy of killing the female larvae. So that's where we are now. We will continue to do this until we no longer detect fluorescence, just as we have for the past 2 years. And we'll do our last slide here. What are we going to do in 2024? And the most accurate thing I can say is we're in discussion. Certainly, FKMCD and the Board of Commissioners would like us to continue. We have questions too. I think we have a lot of complementary questions as to how might our product fit into operational mosquito control? So I don't have an answer. I don't have something definite right now. We are in discussion with FKMCD. That will be a decision that both parties make together. But for now, we are kind of thinking along the lines of how might this fit into an operational mosquito control program? But no final real design has been settled on. So with that, Meredith, back to you.
Thanks very much, Rajiv. Everybody, we are going to dive into our questions now. We have a lot of questions, but feel free to, if you have something that you're curious about that you haven't asked about, put it in the chat box. And again, if you're a Keys resident and can list the key that you live on, we will give priority to those. So we'll start with our first question. Rajiv, sorry, you're not getting much of a break. I think these first two questions are probably best suited for you. But the first question is, were there any observed impacts on the local ecosystem from the release of these mosquitoes?
Well, thank you. I love where that question's coming from. It's coming from a really good place, and I think we share some values about what might the impact be on an environmental stage. Couple things about Aedes aegypti. Aedes aegypti is a non-native species. It is not native to North America or South America or Asia. It is an invasive species, and the other important thing to know about Aedes aegypti is that it exploits temporary water habitats. So while native North American mosquitoes like Aedes taeniorhynchus or Culex nigripalpus do play an important ecological role, they are important sources of nutrition for aquatic predators like frogs and fish and dragonfly naiads. Aedes aegypti does not. So Aedes aegypti larvae develop in temporary, artificial, and natural aquatic habitats. They are not a significant source of nutrition for aquatic predators because we don't find aquatic predators in a bucket or in a wheelbarrow. At the same time, the adult emergence, and we're talking about the local pest population of Aedes aegypti, emerge asynchronously. In other words, you'll get one off, another off, another off. Unlike Aedes taeniorhynchus or Culex nigripalpus, which are highly synchronized, and you will get a large emergence event of Aedes taeniorhynchus, where those mosquitoes can be a source of nutrition for aerial predators like dragonflies. Aedes aegypti as larvae are found in habitats where they do not have predators, and Aedes aegypti as adults emerge in an erratic fashion. They're very small, and they do not emerge in sufficient biomass to act as a nutrition source for aerial predators. And so therefore, there are no predators, aquatic or aerial, that rely on Aedes aegypti. And so we do not expect, and the EPA and FDACS have concluded that the release of our male mosquitoes, remember again, males don't bite, and the subsequent reduction in local Aedes aegypti populations has no impact on other invertebrate or vertebrate predators of insects.
Thanks, Rajiv. This next question will go to you too. And the question is, how many female GM mosquitoes were released or found during the entire 3 years of treatment? So they're asking how many female Oxitec mosquitoes were released or found during the 3 years of the project.
It's easy, 0. And we know that because of those 3 lines of evidence, because we collected eggs, brought them into the lab, hatched them out, collected the larvae, And then we can tell which ones are ours and which ones are local populations. And every single fluorescent larva, which then pupated and emerged as an adult male mosquito. So that's the first line of evidence. The other one is whenever we put out our release boxes, we always have a sister box or a proxy box in a mesh cage. And so we would go in Monday, Wednesday, Friday with a handheld vacuum cleaner and collect the mosquitoes that came out of our release boxes. Every single one for 3 years was male. And the third line of evidence is that we also collected using those light traps. And we collected Aedes taeniorhynchus and Culex and also Aedes aegypti. We certainly collected male and female Aedes aegypti, but when we look at them under the scope, only some of those male Aedes aegypti can contain the fluorescent marker. None of the adult female Aedes aegypti that we collected in the field were ours. And so we have 3 lines of evidence indicating that we did not release any adult female Aedes aegypti.
Thank you, Rajiv. We're going to give you a tiny break. And Chad, this next question is great for you. I'm happy to pitch in if you'd like. But the question is, what role did community engagement play in the success of this project?
Wow. I would say it's paramount. I don't know that I could quantify it, nor would I ever try to quantify it as in a percentage or anything like that. But I would say that a project like this is very reliant upon cooperation and in this case, participation from the public. And I think Meredith and you can kick in here. Almost from the beginning, we've been blown away by the amount. I think the amount of interest we were anticipating, the amount of interest, the amount of questions, and the amount, even concern that we were going to have from the public. And I felt like we spent a lot of time addressing that. But as we got further and further into the project, I think we were more blown away by the amount of people that wanted to be part of it and wanted to participate in it and wanted to learn more about it and further it by actually being a box host or a trap host. At the beginning of the season, there were times where we actually had a list of people that wanted to be part of it. That went beyond what we had in terms of inventory for traps and for box hosts and for that sort of thing. And that's when you participate in a project like this, that's a very good issue to have for a variety of reasons. But I think a lot of people really understood that that this is a project that if it comes to fruition, that it'll be very important. If you're using a mosquito to seek out and reduce the population of other mosquitoes, that to be on the ground floor of that it is pretty important. And I think a lot of people really wanted to be part of it, and we just absolutely been blown away. So I would think next to the actual biology that's involved and the genetics that's involved and the science that's involved, public involvement has just been huge, and it continues to be huge. Even now, it's a great project to talk about because the ramifications as far as human health and preventing illness. I mean, those are all pretty weighty causes, and we're happy to be part of it and happy to talk about it.
Thanks very much, Chad. And yeah, we were in fact oversubscribed all 3 years of the project, and wait lists are a good problem to have. Okay, this next question, I'll take this one. The question is, you are being regulated by the EPA as a biopesticide. That's true. If you want to be regulated as a control for diseases, shouldn't you be asking for FDA oversight? Vaccines have been approved in the US for dengue and yellow fever if you have large outbreaks. Thank you for the question. So our mosquitoes are vector control. They are not disease control. So we are targeting the vectors of disease. We are not targeting the disease. This is all about entomological or insect-related outcomes, and Oxitec does not claim a health impact. Okay, next question, and we appreciate all these questions. Chad, this one can go to you. I think this is probably a good follow-up from what you were just talking about. The question is, how did those who hosted boxes view the project afterwards?
In terms of actually talking to a box host, and I think if I understand the question, it would be, whether or not being a participant was an intrusion on their life, or whether it impacted them as in any sort of negative way. And of course, your folks, Rajiv, the folks that have gone door to door will probably have talked to them a lot more than I have. But the few I have talked about, I never heard a negative impact. I never talked to anybody who it was disappointed with being a trap host or a box host. In fact, usually when I talked to them, they were seeking to get in touch with the correct project personnel to do just the opposite. They wanted to be part of it again, or they were interested, maybe somebody had left a battery or something, and they were wanting to get it was never from a place of negativism. And I do know in the beginning of the project, we talked about, I answered questions from people that wanted to know if there was going to be a cloud of male mosquitoes, or whether or not there was going to be anything that they should be on the lookout for. And I never got any follow-up questions on that. And I think for a lot of the reasons that Rajiv described, their emergence, they're not timed. They're coming out at different times. One thing about the Oxitec solution is they're emergence boxes. So it's not like you're opening a tube and letting thousands of males out at the same time. They're coming out at in kind of a natural rate, and just because of their nature, that's what they do. They come out infrequently. So really a better question for Oxitec, but from a person who answers questions about the project a lot, the interest was always in doing it again, and as opposed to wanting to opt out.
If I may, thank you, Chad. Just to add to that, I'd like to reiterate what Meredith mentioned, which is, well, you mentioned it too. We are oversubscribed. From a scientific perspective, we needed X number of households in a neighborhood, and we typically had 2 or 3X. It got to be such a large number of people signed up as box hosts that we didn't have the manpower to do that. So not only has the support been eager and very cordial, but it's been widespread throughout the Keys. The other aspect, if I can talk about any concern or negativity, it would be from a potential host who said, well, I don't know if I want you to set up a box here. Does that mean that FKMCD is not going to spray ordinarily? And so that was a really good question. It came up often, actually. And I said, great, I'm glad you're asking. And the answer is no. We have designed it in such a way, and we're working with FKMCD so that routine operational mosquito control continues. Because if having a release box in any way interferes with a mosquito control district's tax, tax mandate for mosquito control, this is not going to work. So I'm glad people asked the question, because we could answer that, and I wanted to bring it up tonight as well. No, having a release box, whether it's from a mosquito control district or if you buy it, when it is commercially available, you don't need to coordinate with a mosquito control district, and it will not affect any MCD from doing their routine mosquito control operations.
Thanks, Rajiv. And I'm hoping our webinar won't end automatically in 5 minutes. We've got at least 2 more questions that we would really like to answer. So we are going to try to get those in rapidly. One thing I did want to add really quickly on the box host thing is that we had a number of repeat hosts, especially from this year, from that were carryovers from last year. So that was exciting. The next question is, where may we find the complete field trial data from both trials? So this person is asking about our project, and it's been 3 years now. So eventually we will have 3 years of data, monitoring and some other elements are still ongoing for this 3rd season. But these data are aimed to support a project registration. And at the point of registration, all of that data would be made publicly available by the regulators. So that is something that the regulators review. Our Oxitec team does plan to publish novel findings in scientific journals, which, by definition, those are vetted by third parties. We do not have complete control over when they are published, but these are our plans. Rajiv, if you have anything to add, it's welcome.
Sure. So we don't have a publication on our Florida work yet, and that's going to take a while, right? Because we're not done. We still have our surveillance data to end our 2023 season into 2024. However, right, and 2 things. One, we're going to publish. We'll put that out as a manuscript. It will be publicly available, but that's going to take some time because we have to finish this, and at the same time, we have to include that as a dossier for federal and state regulators. So it is on the agenda, but I cannot give you a timeline. However, we do have publications from our work in Brazil and elsewhere that are available. They are publicly available. And Meredith, you would know better than I where the links are. I think we have links to publicly accessible PDFs on our website. But just wanted to plug that we have published on previous work with mosquitoes in Brazil. We do not have a publication for what we're doing in Florida right now, but one of our objectives is certainly to publish that in a peer-reviewed journal.
Thanks, Rajiv. And the publication that you're talking about from the Brazil data is available on the Oxitec website, and that's something that we're happy to provide in writing after this webinar. And yes, we're still going to answer another question or 2, but if you feel like your question has not been answered, please send it to florida@oxitec.com, and those questions will be answered. So the next question, I'll start to answer this, and Chad, if you would like to chime in, you're welcome to. This seems a little bit more like an Oxitec question, but the question is, are there plans to expand this project to other areas outside the Florida Keys? And there are a couple of things that are important to note here, because right now we are in a regulated project phase. So nothing can be expanded without EPA approvals. We have not applied to expand it at this stage. And if the EPA did register the product, we would love to expand it to other areas where this mosquito is a threat or a problem. But that's our current status. Chad, I don't know if you have anything to add.
No, not really. Just exactly what you said, that we're right now we're concerned with finishing the chapter we're on, but there's certainly some other chapters to be written. It feels like, just because of where we are in the world with mosquito borne illness. So I'm not trying to be a tease, but stay tuned. Just because it's that way right now, we don't know what. We're just finishing the chapter we're on. That looks like there are other chapters to come.
Thank you, Chad. The next question I think would be really good for you to answer too, if you don't mind. The question is, how did you address any concerns of local residents regarding genetic modification?
Well, I think a lot of the problems with genetic modification, I think, comes from just the maybe a little bit of intimidation by the name modification. And I think a lot of it is to begin by talking about how just because something is modified doesn't mean that it's made better or more fierce. In this case, I've always thought from the beginning that a better name for this would have been genetically adjusted or genetically immuted. Because these mosquitoes in many ways are not any more fierce or any more dangerous than anything else, and modified kind of lends itself to believe that. So not all genetic adjustments are created equal. And to point out that what we're talking about here is an emergence of a male mosquito that's basically has an expiration date built into it. And I think once you get into that, and then you point out that the only thing better than our inspectors at finding where female mosquitoes are breeding are an Oxitec male mosquito at finding those breeding mosquitoes. So you're basically using the mosquito against itself, and that really takes care of a lot of those questions. And of course, 3 years of project, a lot of Oxitec males flying around, and no ill effects either on wildlife or on people, really has kind of lent itself to quelling a lot of those concerns. And I think in the Keys, it's natural for people to have concerns whenever you're talking about anything new. People are proud of the Keys, but FKMCD lives here too, and we're very concerned about the environment, but we're also concerned about keeping people safe. And this technology certainly needs to be explored, and that's just where we are. We're in the exploration phase, and let's keep pushing forward.
Thanks very much, Chad. Everybody, we're a little bit over time, but we have 2 more questions that are a little bit different from the questions asked previously. So we are going to get through these next 2, and then we'll wrap up. And a recording will be made available afterwards. So if you can't stay on for the rest, it will be made available to you. Rajiv, I think these last 2 questions are both really good for you, if that's okay. The first one is, how many boxes are distributed to monitor the survival of female mosquitoes, and what is the distribution distance from the home site?
So I'm trying to understand let me answer the second one first, because that's an easy one. The boxes are typically near the home. They're in the backyard, and never very far from the home. So that's an easy one. Maybe, less than 10 meters from an individual house. I'm trying to understand what that first question is. Would you mind repeating it?
Yeah, let me repeat it. Okay, it's the whole question. How many boxes are distributed to monitor the survival of female mosquitoes?
So
And what
how many?
is the distribution distance from the home?
Right,
But yeah, it's kind of a strange question because the boxes are like, we use traps to.
Yeah, maybe. The question itself is a little mixed up. We don't use boxes to monitor the females. We use the boxes to release males. But we have proxy boxes with our boxes, so it's the same eggs, the same water, the same food, except one is in a butterfly cage. And so we typically do that at every site or every other site. So there's good coverage. If the concern of the question is, well, do you have sufficient data to indicate that you're releasing only males, right? You're not making that statement based on one proxy box. That is correct. So it's not at every homeowner's site, but almost every site we have a proxy box in a butterfly cage where we collect the adults, and we can confirm that those adults are male Aedes aegypti, not females.
That's right. And it's the traps and the proxy boxes that are used to detect females in the environment are being released, and not one Oxitec female was found. We talked about that a little bit earlier in the webinar. And the traps have shown us, and I think, Rajiv, you may have presented on this earlier in the webinar, that the dispersal distance is pretty typical for this species. It's about 50 to 100 meters.
Correct. There's nothing exceptional about the flight radius of our males. Other species, so the Aedes taeniorhynchus and Culex nigropelpus, they'll fly for miles. Aedes aegypti typically has very poor nutrition. They don't have a lot of glucose and glycogen, so they don't fly very far. And our male Aedes aegypti fit that profile. They fly about 1 hectare. That's the area that they cover. And so maybe that feeds into the question regarding distribution. Where did you distribute these? We typically distribute a release site every hectare. So it's not multiple houses in 1 hectare. It's one house, one release site per hectare. But we will ask neighbors to help us with trapping. So the light traps and the egg traps, we'll try to get as many as we can, because the more light traps and egg traps we have, the more data we have to understand surveillance.
That's a good point, Rajiv. And, I remember a challenge that we had when we first launched the project is everyone wanted to be a box host. And we need just as many, if not more. I mean, we need really, I think, more trap hosts. And that's a really important component of the project too. And that's something we've, really tried to raise awareness about over the last few years, that, boxes are just part of this, and the traps are one of the most critical components. And, luckily, I mean, the Keys residents, they're all interested in being trap hosts now too. But that was interesting at the start. Everybody wanted a box, but we also need a lot of trap hosts. Okay, last question. This will be for sure the last one. We're about 10 minutes over. Rajiv, I think this is good for you. The question is, how many homes participated, and what was the average percentage of participants in a neighborhood?
Oh, that's a great question. Well, the answer to that is it depends on the year. a few hundred hosts? So the answer is hundreds for light traps and oviposition or egg traps. So we always had more people who we needed to help with the trapping. So that was a really good coverage. That was in the hundreds. The number of homeowners for box release, like I said, we only needed one home per hectare. So for example, if there was, if it was a 12 hectare neighborhood, we only needed 12 people in that. So overall, I would say less than 100 actual box hosts, but hundreds of light and egg trap hosts.
Thanks very much, Rajiv. And yeah, I mean, I think every year when you add up the various traps that you all saw that we've used as part of this project, and also the boxes, it's been over 500 homes and in excess of 1,500 residents for each season. And there have been 3 seasons now. So incredible support, and we're really thankful for that. We are going to wrap up the webinar now. Again, if you feel like your question hasn't been answered, I mean, we tried to get to everything, and I think we did. But please send that question to florida@oxitec.com. We will respond, and the full recording of this webinar, along with the slides, will be posted to the project website, keysmosquitoproject.com, in the coming days. But that is all from us. Please join us for the next webinar, and thank you all.